So let's look at gas transport. We've got this gas in, and we're primarily just talking about oxygen and CO2, right? We've got this gas in. Now, how do we get that gas to where we need it to go? Well, with this, we are talking about internal respiration, which is something that they don't call it anymore. They just refer to it as systemic gas exchange, okay? So that's how I'll reference it, on the exam, systemic gas exchange. We have carbon dioxide and we have oxygen. Remember, carbon dioxide has a very high solubility coefficient. Oxygen does not, okay? You carbonate your drinks. You don't oxygenate your drinks. The amount of pressure, amount of force that we have to get carbon dioxide into a liquid doesn't take much. For oxygen to get into a liquid, it requires a hell of a lot of pressure. So it's not done. So because carbon dioxide can easily be picked up by fluids, water, the plasma, blood, we're going to see some unique characteristics associated with that compared with oxygen. Okay, so carbon dioxide moves from systemic cells into the blood. Moves from systemic cells into the blood. Why is it doing that? We have an increase in CO2 because we have metabolic activities, occurring within the cells. And one of the byproducts of metabolism was an increased production of carbon dioxide. And so it's going to be built up here in the cells as they are doing the work that cells normally do. And remember, we go from high concentration to low concentration, or high pressure gradients to low pressure gradients. Pressure gradients, concentration gradients, guys, they are the same damn thing, at least for us right now. Think of it in the same. Okay, everything goes from high to low. So this is no different. So we have high carbon dioxide in the systemic cells. We have low concentration right here, as the blood is great arrow right here, shows the blood's going in this direction. Okay, blood's going in this direction. So as that blood enters this region with where the systemic cell is, the CO2 is going to go from high to low. From high to low. All right, and this diffusion continues until, it reaches equilibrium. Okay. Everybody with me there? Because blood is not very soluble because oxygen is not very soluble in blood, it binds to the iron of hemoglobin. The vast majority, and 98% would be the vast majority, That's why iron is present in hemoglobin. That's why we have hemoglobin, because that's going to carry oxygen. Oxygen doesn't bind to anything else. Oxygen doesn't go into the plasma in any significance, okay? When oxygen is bound to hemoglobin, we refer to it as oxyhemoglobin. When hemoglobin has no oxygen bound to it, it's referred to as deoxyhemoglobin. Oxyhemoglobin, as it goes through here and dumps off oxygen, deoxyhemoglobin. And we can determine our hemoglobin saturation levels by reading the difference, the ratio between oxyhemoglobin and deoxyhemoglobin. So with oxygen, it's pretty straightforward. It just winds up on the red blood cell, specifically hemoglobin, iron of hemoglobin. When it comes to CO2, because it has such a high solubility, the vast majority of it is going to be dissolved in the plasma. Okay? And I'm not going to ask you the percentage, but the vast majority of carbon dioxide is transported in the plasma as bicarbonate. We get some CO2 that's dissolved in the plasma. So there's a whole lot of this going into the plasma, right? And some of it will attach to the amino group or the amine group found in hemoglobin. Okay, the amine group found on the globulin portion, not iron. Okay, surrounding it. So we got a little bit that gets transported on a red blood cell. You get a little bit of CO2 within the plasma portion of blood. And the rest of it gets traveled around as bicarbonate. And here you can see how we get this bicarbonate. Okay, when we start to pick up CO2 by the amine group of the globulin of hemoglobin, it combines with water, forms H2CO3, and carbonic acid is detrimental. So chloride ion comes in. That helps us to exchange this here, and the enzyme carbonic anhydrase ultimately is going to help us break this carbonic acid up. We get bicarbonate ion and a hydrogen ion. Okay. I need to get this out. I want to get this bicarbonate ion out of the red blood cell and into the plasma because that's where it likes to be. Okay. Everything has to be in balance and equilibrium. So I have to have a negative ion come in to have a negative ion come out. Okay. So bicarbonate ion comes out, chloride ion comes in. I've maintained the pH balance of everybody, and everybody is happy. And the great thing about that, HCO3, I can use that to help maintain pH balance of the fluids. I can break it down into CO2 and water again. I can do this in the lungs. I can do this in the kidneys. I can do this any place where I can regulate the presence of stuff, the lungs and the kidneys. As bicarbonate ion dissolves in the plasma, CO2 diffuses into the erythrocyte there, okay, like I just talked about, combines with water, ultimately forming bicarbonate. Bicarbonate diffuses into plasma. CO2 is regenerated when blood moves through the pulmonary capillaries and the process is reversed. So this was over here into the system. This is in the pulmonary circuit. Here's bicarbonate ion floating around. Well, I can't just pass through here. I got to get into the red blood cell, be converted back. Carbonic anhydrase helps again to break down H2CO3 carbonic acid into water and CO2. And now CO2 can easily diffuse out. This is not CO2. HCO3 is bicarbonate ion. Bicarbonate ion is not exhaled. Bring it in, chloride comes back out, bicarbonate goes back in, reverse the process, and voila, CO2 exhaled. This is fabulous because, again, like I said just a few seconds ago, bicarbonate is an important buffer throughout the system, throughout the body. And its presence there within the plasma of blood means we can easily convert it back and forth depending upon what we need to do with the pH of the blood, pH of the body. It is very interesting when oxygen binds to a red blood cell, stuff changes. And think of a red blood cell like a bus. As soon as one oxygen molecule binds to it, more people want to jump on board, like a bandwagon, right? As soon as somebody jumps on and things start looking up, everybody wants to join. Okay. Well, oxygen again is binding to the iron, which is in the middle of a hemoglobin molecule. Carbon dioxide and the hydrogen ions bind around it. But when it comes to oxygen, we get a slight change in shape. Okay. So when oxygen binds, it's going to change its shape a little bit. Not something along the outside where it's going to look all weird and deformed like sickle cell, but internally. Okay. Internally, the structure starts to just change a little bit. This makes it more readily for more oxygen to get on. As more oxygens load up, it's going to continue to have just a slight kind of shift in its molecular shape on the inside. Eventually, it gets to the point where it's like, I'm full. I have no more seats on this bus, and I cannot load any more oxygen. And that molecular change on the inside is what really prevents oxygen from binding excessively onto red blood cells. There's only a certain amount of oxygen molecules that can bind. I don't know, something like a billion or something. I don't know. It's like a crazy number of oxygen molecules that can bind. This oxygen hemoglobin saturation curve kind of highlights that. Okay? If the percent as the percent oxidation increases, the number of red blood cells or the number of oxygen molecules binding onto a red blood cell increases. And again, there's a point where it reaches saturation and nobody else can get on this thing. Excuse me for a second. Sorry about that. Okay. So if we look at this curve here, what do we see? Well, at the very beginning, we have a very steep curve. So just a small change in saturation, we get a huge bump in oxygen wanting to get on board. And this is pretty steep to about, 75% saturation. That's not a huge amount of partial pressure. That's not a huge amount of oxygen molecules. But the little bit that's there, boom, easily binds up. After about 90% saturation, the bus axles are like, dude, I cannot get any more of you guys on here. And as you can see, this curve starts to really flatten out quite a bit compared to what it did here at the beginning. So the percent saturation is greatest when you have less oxygen available. The percent saturation is greatest when you have the smallest amount of partial pressure of oxygen available. So when you're at higher altitudes and there's less oxygen, it is easier for oxygen to get onto your red blood cells. Oxygen is less as you get higher in atmosphere. So the body's response to that is to allow this to quickly happen. The body's also responding to low oxygen. It's going to stimulate EPO production, and that's going to stimulate red blood cell production. So you can increase your red blood cell production when you go in a higher altitude, right? increased red blood cell production because we are looking for all the oxygen as possible. So again, large changes in saturation occur with small increases of partial pressure of oxygen. Okay, when it's at lower partial pressures, this curve is really steep. Okay, and really by about 98% percent saturation is about 100%. It goes over, slightly over. It's 104, and that's where your 104 millimeters of mercury comes in. They're in the lungs. So you can figure out your percent saturation by understanding where you are at altitude. I'm not going to ask anything from this slide, so don't worry about it. Are there other things that can happen besides a change in pressure that can affect a red blood cell's ability to grab onto oxygen? Oh, heck, of course. Partial pressure gradients, Dalton's law. All of it is based on a constant temperature. So what happens if we change that temperature? If you elevate temperature, it actually makes it easier for hemoglobin to get off. It diminishes hemoglobin's hold on oxygen. What happens when you're exercising? When you're exercising, you increase the temperature in the muscles. Okay? You're also increasing metabolic demand. This makes it super fantastically easy for hemoglobin to unhitch itself from the red blood cell and get into the tissue. As we go into these tissues where temperature is increased and metabolic activity is increased, what has also happened? Well, you're increasing metabolism, so you're increasing hydrogen ions, you're increasing CO2, all that fun stuff. As we get into there, I've easily dumped off oxygen. Oxygen easily has removed itself from hemoglobin. As these easily hop off, hydrogen is going to start binding to hemoglobin. And it's going to make a change, conformational change to that structure. As this structure changes, all of those oxygens are like, oh my God, I'm out of here. Because we're changing the shape of it and it doesn't want to stay attached to something like that. So they jump off and they don't want to come back. So this decreases the affinity for oxygen to bind to hemoglobin. Oxygen releases and gets into the tissue where a need is obviously required. Excuse me. This is called the Bohr effect. As temperature increases, the affinity for oxygen to stay bound to hemoglobin diminishes greatly. So it releases off of the hemoglobin, releases off of the red blood cell, and gets into the tissue. As that is happening, hydrogen ions are going to bind to it. We get a release of CO2 into the plasma. CO2 gets picked up by the red blood cell, gets converted into bicarbonate ion, and we get an increase in hydrogen ions. And this is called the Bohr effect. All because of that slight little conformational change that has occurred. There's something called 2,3-BPG, and you can see that listed right here. Okay. 2,3-bisphosphoglycerate. It's a molecule that's only found on red blood cells. It helps to, it makes it easy for it also makes it easy, sorry about that. It also makes it easy, 2,3-BPG. Also makes it very easy for red blood cells to release oxygen as well. This is kind of significant because you see a greater concentration of 2,3-BPG in red blood cells associated with growth hormone and testosterone, thyroid hormone. Those are the most common ones. Who has a lot of testosterone? Boys. Where's all that oxygen going? Muscles. Greater musculature is associated with men because of the increase in testosterone. Increase in testosterone does a number of different things. One of them is to decrease the affinity for oxygen to stay bound to red blood cells. So it wants to get into the muscles, and you can do more work, get bigger muscles. This also occurs again with thyroid hormone. Thyroid hormone is associated with an increase in metabolism, which is again associated with growth. CO2 can affect the binding. Okay, CO2 can affect the binding. Where'd my pen go? Okay, CO2 can affect the binding of oxygen to hemoglobin. As more CO2 binds to hemoglobin, oxygen doesn't want to be associated with that. So it's going to want to offload. So the binding of CO2 helps to decrease the affinity for oxygen to stay bound to hemoglobin. And oxygen bounds off and gets into the tissues. So we have partial pressure gradients moving these things. We have temperature moving these things. We have the Bohr effect, which increase in hydrogen ion concentration because an increase in CO2 production, 2,3-BPG, and just plain old CO2 binding to it. So there are a number of things that can affect hemoglobin's ability to stay bound to oxygen. Carbon monoxide even. CO2 easily wants to be in the plasma, okay? But carbon monoxide loves to stay bound to hemoglobin, okay? It has a high affinity for it, so much so that it actually prevents oxygen absorption into the red blood cell. And that's why carbon monoxide poisoning is so deadly, because it prevents oxygen from binding to hemoglobin, therefore decrease in oxygen, therefore death. I'm not going to ask you to interpret the curve, so don't worry about it. But what I love you to know about temperature and the partial pressure gradient that we talked about earlier, and the Bohr effect and 2,3-BPG and CO2 binding, sure, they make fabulous multiple choice questions. Maybe a short answer, but I don't know how I'm doing it this semester.